Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 33. The topic is carbohydrates. Discussing agenda for the topic is introduction, monosaccharides and disaccharides. Lecture outcome. After watching and listening this lecture, students will know about the structure and different types of carbohydrates. Introduction. Carbohydrates serve as fuel and building material. Carbohydrates include sugar and polymers of sugar. The simplest carbohydrates are the monosaccharides or simple sugars. These are the monomers from which more complex carbohydrates are built. So, monosaccharides are the building blocks of other complex carbohydrates. Disaccharides are double sugars, means two monosaccharides combine with the help of a glycosidic linkage, and the structure formed is called a disaccharide. And when the number of monomers increases beyond 10, then we called such a carbohydrate as polysaccharide. Now we will discuss the three major types of carbohydrates one by one. The first is monosaccharides, which are the simple sugars. The Greek monos means single and saccharide means sugar. So these are the simple sugars and their chemical formula is CH2O, or the multiples of this formula. Means glucose is a simple monosaccharide. Its formula is C6H12O6, and another sugar, which is the table sugar commonly consumed in tea or other sweeteners, its formula is C12H22O11. So the simple sugars has the formula which is a multiple of this unit CH2O. The glucose is the most common monosaccharide, and it is of central importance in the chemistry of life. Remember, glycolysis, which is thought to be the most ancient energy deriving pathway, and the starting molecule for the process of glycolysis is glucose. In the structure of glucose, you can see the trademarks of a monosaccharide. What are the trademarks of monosaccharide? A carbonyl group and multiple hydroxyl groups. These are the trademarks of the monosaccharides. Now we can further classify monosaccharides, and there is certain criteria for this classification. The first is the number of carbon atoms in monosaccharides, which can be 3 or 4 or 5 or 6. So the trioses have 3 carbon atoms, The pentoses have 5 carbon atoms and the hexoses have 6 carbon atoms. Another classifying criteria is the position of the carbonyl group. If the carbonyl group is present at one end, then such type of a monosaccharide is called aldose. If the carbonyl group is present within the carbon skeleton, then this kind of sugar is called ketose. So remember the difference between aldose and ketose. The classification is based upon the position of the carbonyl group. Glucose is, for example, an aldose, and glucose is the isomer of fructose. And fructose is a ketose sugar. So remember, glucose is an aldose sugar and fructose is a ketose sugar, and both glucose and fructose are isomers. You can see here glucose, the carbonyl group is terminal, so it is an aldose sugar. In fructose, the carbonyl group is in the middle of the chain, so this is a ketose sugar. Now most names for sugars end in the phrase "ose". Another criteria for classifying monosaccharides is the size of the carbon skeleton. We have already discussed that the number of carbons in the skeleton can range from 3 to 7. Glucose, fructose, and other sugars that have 6 carbons are called hexoses. Trioses and pentoses are the ones having 3 and 5 carbons respectively. So here what you need to remember is that glucose is a hexose sugar, and the pentose sugar of DNA and RNA is one of the types of the monosaccharides. Still another source of diversity for simple sugars is the way their parts are arranged spatially around asymmetric carbon. Asymmetric carbon is that carbon which has different partners attached to it. Glucose and galactose, for example, differ only in the placement of parts around one asymmetric carbon. You can see here glucose and galactose. So they differ in the arrangement of the parts around the asymmetric carbon. You can see the difference here. There is a shift in the position of the hydroxyl group in both the isomers. What seems like a small difference is significant enough to give the two sugars distinctive shapes and binding activities, thus different behaviors. So this difference seems to be a minor one, but it has significant effect on the structure of those sugars and thereby on the characteristics and properties of those sugars. Here we will analyze it in detail. Although it is convenient to draw glucose in a linear carbon skeleton, this is a linear carbon skeleton, but because inside the living organisms there is always an aqueous environment, so the hexoses and the pentoses, they have a tendency to form a ring structure inside the living organisms, in the aqueous solution. You can see here, this is the linear carbon skeleton, and in a reversible reaction it is forming a 6-membered ring structure. Monosaccharides, particularly glucose, are major nutrients for cells because these glucose molecules are incorporated in the glycolysis and thereby yielding energy for the cell. In the process known as cellular respiration, cell extract energy from glucose molecules by breaking them down in a series of reactions. Not only are monosaccharides a major fuel for cellular work, but their carbon skeletons also serve as raw material for the synthesis of other types of small organic molecules such as amino acids and fatty acids. So the monosaccharides, they not only provide energy, but their carbon skeleton acts like a precursors for the synthesis of amino acids and fatty acids. Monosaccharides that are not immediately used in these ways are generally incorporated as monomers into disaccharides or polysaccharides. So just summarize the monosaccharides. Monosaccharides are the simple sugars. The number of carbon atoms can range from 3 to 7. The monosaccharides are classified on the basis of the number of carbon atoms in the carbon skeleton, or on the basis of the position of the carbonyl group, whether it is terminal present at the end of the carbon skeleton or in the middle of the carbon skeleton. And the prime use of monosaccharides is to yield energy. The glucose molecules are incorporated into glycolysis and ATPs are produced. The carbon skeleton of monosaccharides is used as a precursor for the synthesis of amino acids and fatty acids, and still the monosaccharides can combine and form disaccharides and complex carbohydrates or polysaccharides. Now disaccharides. Disaccharides consist of two monosaccharides joined by glycosidic linkage. Glycosidic linkage is a covalent bond formed between two monosaccharides, and just like other polymers, the reaction which combines monosaccharides or which combines the monomers to form a polymer is called dehydration reaction, in which one of the water molecules is eliminated. For example, maltose is a disaccharide formed by the linking of two molecules of glucose, also known as malt sugar. Maltose is an ingredient used in brewing beer. The most prevalent disaccharide is sucrose or table sugar. Its two monomers are glucose and fructose. Remember, sucrose is the table sugar and it is a disaccharide, and the monomers of sucrose are glucose and fructose. Plants generally transport carbohydrates from leaves to roots and other non-photosynthetic organs in the form of sucrose. Lactose is another disaccharide which is present in milk, and the two monosaccharides from which lactose is formed are glucose and galactose. Now here is a interpretation of the chemical reactions which forms maltose and sucrose. So maltose is formed from two glucose monosaccharides. The chemical reaction which takes place here is condensation or dehydration, in which a water molecule is removed and glycosidic linkage is established between the carbon 1 of one sugar and the carbon 4 of another sugar. So this linkage is 1,4 linkage. Similarly, the sucrose or the table sugar is formed when two monosaccharides, glucose and fructose, combine in the chemical reaction is same. The water molecule is removed. Now the disaccharides must be broken down into monosaccharides to be used for energy by organisms. The organisms cannot yield energy directly from disaccharides, so they must be broken down into simple sugars. And in the last slide we talked about lactose, which is present in milk. Now lactose intolerance is common in humans. Some humans, they lack an enzyme called lactase, and therefore they are unable to break down the lactose present in milk. Now what happens in those cases? The sugar for them is broken down by the intestinal bacteria, which causes the formation of gas and crampings. The problem can be avoided by taking enzyme lactase when eating or drinking dairy products or consuming dairy products that have already been treated with lactase to break down the lactose. So just to summarize that, the humans cannot use disaccharides for energy. They must be broken down into their individual monomers in order to be used as a source of energy. Lactose is present in milk, but some individuals in the population are unable to break down lactose because those individuals lack the lactase enzyme, which is used for breaking down the lactose. So in those individuals, the gut bacteria break down the lactose, and as a result, the gas is accumulated, and the person suffers from gas trouble and cramping pain in the belly area. This problem can be avoided by consuming synthetic lactase or by consuming dairy products which are enriched with lactase enzyme. This was the end of the lecture. In the next lecture, we will discuss the third type of carbohydrates, which is polysaccharides. Thank you.